work done by Craig Reynolds. So what Craig did was he used intuition to think about what types of rules that individuals use in groups. And so he assumed that they repel locally, and they're attracted and they align with each other over longer distances. And this is back in 1986. Remarkable. He showed that you don't need a leader. This is distributed control. And of course, now we can use GPU computing to go way beyond this, both in terms of the number of individuals we can simulate, and what's equally important, I mean, it looks less impressive on the screen than this wonderful simulation by Bernardino Frollo, who's here today, and his collaborators and myself. We can also run smaller simulations, but have hundreds or even thousands of them running concurrently. So we can explore parameter space in ways that weren't possible before. And, you know, I find it difficult to do programming if I have to leave something running for 3 weeks and then come back and look at the results. I don't relate to my software in the same way. I don't relate to science in the same way as I can if I can run it, go and have a coffee, and come back and get those results and visualize them. So for me, the whole way I do science has been transformed by GPU computing. Okay, so I'm going to return to using an example of fish, okay, but think much more broadly than this. I have these nice pictures of fish from Christoph Becker. And what we can do is we can represent individuals existing at certain positions, which is represented by C, the centroid or the center of gravity of that individual, and going in certain directions. But of course these individuals interact, and we can simulate these interactions using very simple equations that represent the general tendencies. For example, a good tendency to avoid colliding with others if you get too close. For birds, collision can actually be fatal. So this is the highest priority rule that the organisms tend to use. If individuals become isolated from each other, however, it's dangerous, and I'll show you some of the risks of predation later on. And so what do individuals do? Well, they tend to be attracted towards each other, a form of biosocial attraction, which is what keeps groups together, and they can tend to align their direction of travel, which means they tend to move in the same direction as their neighbors. And so this is the attraction term in the equation, and this is that tendency to align. And what organisms can do, because they're not particles, they're organisms, they can change behaviors in response to external conditions, and they're using, for example, the visual system to mediate these interactions. They can change these rules dynamically and in a context-dependent fashion. And so I'm going to show you how we can use computation to scale up from these types of interactions to the sorts of dynamics we see at the group level. So here is the same simulation, and it's in 3 dimensions. So the arrow is representing the direction of travel of the individual. The ZOR is that zone of repulsion that I told you about, where you tend to avoid others that get too close. The ZOA, which is the outer zone, is the zone of attraction. If you get isolated from others, you will tend to veer back towards them. And in between, there may or there may not be this local tendency to align your direction of travel with near neighbors. And the way that alignment comes is system specific. For example, if you're dealing with cells who are moving together, then adhesion molecules on the surface of the cells create frictional forces as they're passing each other, which causes a tendency to align. You'll see in locusts later, there's no explicit alignment, yet they align. And so we're not saying where these functions necessarily come from, but that individuals follow these local rules. And if we explore the model where we just have repulsion and attraction, so local repulsion and longer range attraction, we see here, this is a static image of course from a mobile simulation, we see here a sort of mosquito-like swarm. And what I'm going to do now is introduce that tendency to align your direction of travel. And we see that this creates a sudden change in the group dynamic. Now the group is forming what we call a torus, where they're perpetually rotating around this empty core. But there was nothing in those equations of motion that said go in a circle. So we couldn't have thought through how these interactions lead to collective behaviors. The computation has to guide us. And for exactly the same region of parameter space, for exactly the same individual interactions, if we run the model for long periods of time, we not only see this torus formation, we also see what we will typically think of as a fish school or a bird flock, where the individuals are in this sort of living fluid that moves through space and individuals can change positions. And so the model is useful. It makes a fundamental prediction that there are these three sort of states of matter, so to speak, in terms of the collective dynamics of these types of groups. And that the transitions between them are very sudden. And this is something that people in the physics community are very familiar with. You know, there are lots of different molecular compositions, and yet you see liquids, solids, gases, plasma, and so on. And so this model is predicting that similarly, when you look at what are called fast equilibrium systems, systems where we're not adding thermal energy to excite the system, they're actually adding energy themselves, we predict these three fundamental states. And so when I went to Princeton in 2008, I wanted to try to test these ideas. Can we build an experimental framework that can allow us to get the same level of detail as we previously had in models? And here you can see a video of a small fish called the golden shiner swimming in a tank that's 7 foot by 4 foot with very shallow water. These fish love to swim in a two-dimensional type environment, which is very fortuitous because 3D tracking is a real pain. In fact, even the 2D tracking problem is fairly difficult. With Haishen Wu, a collaborator in China, we've developed some software, well really he has developed software, that allows us to track the motion of these fish, and it can deal with problems such as occlusion of near-identical individuals, which is the core challenge here. So this allows us to really get inside the heads of these individuals to understand how are they interacting. And here I'm showing you tracking one individual, but of course we track lots of individuals, and I'll show you that again shortly. And in all of our many hours, tens of hours, perhaps even over 100 hours of tracking these fish and recording their behaviors with different group sizes and different arenas and so forth, we only ever have seen those three fundamental states that were predicted by the model: the swarm, the torus, and the parallel group state. And you can see these hot spots in that figure at the bottom, which shows you the degree of rotation, which is actually the angular momentum of the group, which is the torus formation. And when that is low and the polarization is high, that's when we have this moving fluid-like swarm. And in the bottom corner there, that's the swarm behavior. And they will stochastically flip between these different states in a sort of meta-stable regime. But I said, we can track not just one individual, we can track hundreds or even more individuals. We can get their positions, their head position, their velocities. But what we also want to do is reconstruct what they see. So again, I mentioned this before. Fortunately for us, these fish coordinate their behavior by visual means. And so we can track, so with Colin Toomey and Bernardino, we can track the positions of the eyes of the fish and then use raycasting algorithms to reconstruct the visual field. In fact, we've cut down massively the number of rays cast in this image just to make it look pretty. In reality, we can project very, very large number of rays. And we also not only want to do this in a sort of post-processing manner, we also want to do this in real time. So we can create perturbations to the group when individuals have certain types of information, or when, as you'll see shortly, the network topology of the information flow is in certain regions of interest to us. And the way we can do that is by using OptiX to actually track the motion of fish in real time, calculate the positions of the eyes. We also calculate the structure of the body, which we also think is important, so we can really see the world from the perspective of the individuals. And this then allows us to reconstruct what you see here, which is a sensory network. The edges between these fish, this is not a simulation, these are real data. The edges between these fish represent the area subtended on the retina of other individuals. We can also represent these edges to mean the optical flow on the retina, or other features of biological interest. And I'll show you this movie again, because here there's a perturbation, an individual gets startled, and we're interested in how does this information percolate through the network. There's lots we don't know about biological networks and how information flows. So again, we think these fish are a very nice model system to understand these types of dynamics. And why are we interested in these organisms, these types of organisms? Well, look here at the remarkable collective properties of these animals within groups. Look at them responding to predators. You can see why in the 1940s and the 1950s, people believed that they used telepathy or thoughts transference to coordinate their behaviors. But this is the same model I showed you before, and I've added one more rule, which is a biologically plausible one. If you see that predator, you try to escape. And so lots of the patterns that we see in nature can be explained by simple, relatively local rules of interaction on the part of the individuals. And so natural selection is very likely to find these types of behaviors. You can see a vacuum around the predator here as it enters the group, trying to find the prey. Predators can also be confused by the prey within these types of groups. So we're not only interested in trying to understand the prey's perspective, we also want to understand the predator's perspective. And again, I want to reiterate that this is distributed control. There's no leader, there's no hierarchy. It's distributed control. And I love leaving it to penguins. I mean, look at that, isn't that absolutely stunning? They look so ungainly on land and so stunningly beautiful under the water. And so one way that we can try to get inside the head of the predators, we're actually not really allowed to work with real predators these days for ethical reasons. So what we've developed with Naomi Leonard and Dan Swain is Robo-Predator. So this predator, again, we can track it in real time. We have a closed feedback loop. There's an autonomous robot underneath the tank, controlled to this predator model using magnetism. So that's why you can't see the robot underneath. But of course, this predator can also see the world. We can use raycasting to let it see the prey. It can also make its own decisions where to attack and when to attack. So we can begin to get insights into the evolution of good strategies for dealing with collective dynamics. We also study in the field. So I'll show you some pictures from Mauritania later on. But this is a field study that's just been accepted for publication, where we've used a new type of sonar technology called DIDSIN. And what I'm so excited about with DIDSIN is that it has a high temporal resolution, but also, and I think this is amazing, also it's a sonar with a 2 centimeter spatial resolution. And so what we've done with Kevin Boswell, our collaborator, is to take DIDSIN and actually film in towards the shore. So I'm going to show you these kind of triangular-like pictures, but this is not us looking from the surface to the depths of the ocean. It's actually looking down from above to this type of cone. And the water depth is only 1 meter. And look at these beautiful pictures of real predators attacking real prey under natural conditions. And this is in an estuarine environment in Louisiana, which is at risk. So there's lots of very important ecological reasons why we want to survey animals in this region, and yet nobody's been able to survey with this type of resolution before. And how can we do this? How can we maintain long-term surveys in areas like this? Well, we can use computing to analyze these images in real time to provide a stream of extremely valuable data about the predator-prey interactions within this at-risk habitat. So for example, if there are oil spills or other perturbations, we now have the technology in place to quantify what impact that has on these key species. And one of the themes that really percolate through my research, and I think is a very exciting direction in which we're trying to push our research, is trying to understand, can we think of these groups as some sort of collective mind? I told you before, there isn't telepathy within these groups, yet there clearly is some form of information propagation and information processing that occurs at the collective level. How can we relate individual level cognition to collective cognition within groups? And so one way we can begin to gain insight is to use computation. So I'll show you some work we've been doing on leadership within groups. So leadership, again, is something fundamental not only to human society, but also to animal groups and collective decision-making. How do these types of animals, or even cells, make collective consensus decisions? So first I'll start off with leadership. I mentioned before that they're not particles. Individuals have memory. They recognize, you may think fish have an 8-second memory span or whatever the popular press wants you to believe. That's absolute nonsense. It's a myth. They can remember for weeks, for months, for years. incredibly good memory. And we rely on this to do our experiments. So this individual here may have remembered that there's a food source over there. The others may not be aware of it. Similarly, in our societies, different individuals have different types of information. Does it have to signal? I know where to go, follow me. The other individuals need to recognize who has and who does not have information. Does that even make sense in evolutionary terms? These are selfish, unrelated individuals. Why would they pay a cost to signal to others this type of information? So using a very simple two-dimensional model, we always try to use the simplest possible model to explain these phenomena. These results also hold for more complex, multi-dimensional models. But here we have an individual responding socially to its neighbor, with a vector that we call S for that social tendency, that schooling tendency. But if the individual has information, I'm coloring it in bright red. Now what's really important to remember and to know is that the individuals cannot tell who has information and who does not. These colors are only so we can see. Okay, so whenever I'm showing you colors, it's so we can see the informational status of individuals. There's no cheating going on, they've got no way of inferring this. They start at random positions and random orientations. But individuals that do have information, this goal-oriented tendency, G, have to reconcile that with their social tendency. And we assume they do so with a parameter. This is another important thing to remember. A parameter that we call omega, that I've shown in green there. And what that omega really is how strongly you weight your goal-oriented information. If that weighting is relatively low, you're not really hungry, you don't really need to go over there, and so it doesn't have a large impact on your motion. However, if omega becomes large, then your goal-oriented behavior becomes very important to you. You really want to get your own way and go in that direction. And so one of the first things we can ask is, can information percolate in groups without signaling and without individual recognition? And so I'm going to show you 3 movies where the informed individuals, again for our benefit, are going to be colored white, the uninformed are going to be green, and the big green cross is just going to show you the center of gravity of the group. And I'm going to show a group of 100 being guided by 1 informed individual, 5 informed individuals, and 10 informed individuals. And so if we look first at 1 informed individual, you can see it's not really able to guide the group in its preferred direction. Whereas if we look at 5 informed individuals, after an initial period of reorganization, you can see that information gets propagated and the group, kind of moves in the right direction. And if we have 10, then suddenly and spontaneously the information is transferred and the group moves very accurately. But, this is nice to look at these pictures, but we also want to quantify, to measure how accurately was that information transferred. And that's what this figure here shows us for different group sizes. So let me take you through the key message to be drawn from this figure. On the x-axis, we have the proportion of informed individuals within each group. The different colors represent different group sizes. The black line being the lowest, the smallest group size of 10, the red line being the largest of 200. The y-axis now measures the accuracy of information transfer. From 0 being completely random, so it didn't transfer at all, to 1 being perfect. And of course, these are local, noisy little creatures, or simulated creatures in this case. And so they never quite reach that perfect information transfer capability. Okay, so now we can look, say we want a group to move at 85% accuracy, I've arbitrarily chosen that. Now for a group of 10, for that level of accuracy, we need approximately 50% of the individuals to have information. But if we look at a group of 200, the red line here, for the same level of accuracy, we now need less than 5% of the individuals to have information. So as group size gets larger, the proportion of informed individuals you need to guide the group gets much, much smaller. And this is an important result that we'll return to later, where we're trying to understand the evolution of leadership in nature. Because this tells us something about natural systems, such as these herding caribou, or migrating birds, and perhaps importantly, or most importantly, migrating cells. Here you're seeing an image of a brain tumor invading, and you can see cells stained in red. These are leader cells that are pushing through the tissue secreting a chemical called protoplanin that causes calcium oscillations at the onset of collective migration. We want to try to understand why did we get leadership in nature from bird flocks to cancer cells. And that's where computation and trying to understand the evolution of this type of dynamic is important. And that's where CUDA also comes in as extremely helpful. Because we don't want to simulate 100 individuals. We don't want to simulate 200 individuals. We want to simulate tens of thousands or hundreds of thousands of individuals moving through complex environments, such as the spatio-temporally varying resource landscape, which could be a thermal gradient in the ocean, for example. We not only want to do this, here you're seeing them responding over an ecological or a behavioral timescale. We actually want to evolve them. You saw the wonderful example in Jensen's talk of galaxies colliding and looking into the future. But we also want to look into the future. We want to evolve populations and ask, how does human modification of the environment impact migrating organisms? We want to ask, how can chemotherapeutic agents impact migrating tumors? And this is what CUDA has been really essential for. Because here on the x-axis, we're now not looking at ecological time, we're looking at evolutionary time. We're looking at generations. So we can allow these virtual organisms to live their lives, to reproduce, and to evolve. And that's not been possible before. And so here you can see something interesting. If we allow these populations to evolve, they evolve to detect the gradient in the environment. So for example, in birds, this could be the magnetic field. In humans, this could be a glucose gradient. But it's costly to do that. And so individuals then begin to exploit those that detect it. So initially, they evolve social interactions because they can help each other out in this noisy environment. But that's not evolutionarily stable. And you get what's called an evolutionary branching process, where the population evolves into leaders, which is that little peak, and followers, which is the big peak. The vast majority of individuals are using social information to guide their motion. And we think this occurs in lots of different systems. It's a very robust result. I also wanted to mention two other areas where computation is really important, is we're also able to evolve individuals within very complex, spatio-temporally very complex environments, such as fluids. Now again, we couldn't do this before CUDA. We're also interested in testing our ideas. So here at the top you see fish navigating a light gradient. They love to be in the dark regions. In the lower image, it's exactly the same scene, but filmed using infrared light, which allows us then to map the positions of the real fish to the gradient that they're trying to detect. And I can't talk about this work now, it's not published yet, but it's extremely exciting, the algorithm that the fish taught us by how collective navigation works. So far, all of my leaders, all of these informed individuals, have been wanting to move in the same direction. They've been in agreement with each other. Well, of course, we know that often there can be disagreement within groups, and that's the same within animals. So this now represents the case where the red individuals want to go one way, and the green individuals want to go another way. Can the group come to a collective consensus decision? So say we all decided we wanted to go to the same bar tonight. And I don't know San Jose, so I ask you, does anyone know a good bar? I say, I don't know, 10 of you say that there's this great bar down the road. And 11 of you say, no, there's another really good bar. How do we decide which one to do if we all want to do it together? You know, I do a little bit of math and I pass back, well, the majority says do this. But of course in these groups, they don't have that capacity. They cannot talk to each other. And what I'll show you is they don't need to. Using these simple rules of interaction, these groups, so here there are some individuals that want to get to the white target and the red target, 10 to each respectively. And in this case, they randomly choose one, the red target. But if I add just more one white individual, and I hope you sort of see the local, noisy nature of the interactions in this movie, over 99% of the time, they will accurately choose the majority direction. They cannot count. They are solving this through the kinematics, through the motion of their bodies and their interactions. So I find really remarkable. But of course, individuals can be stubborn and unwilling to give up on their opinions. So if that omega parameter is large enough, then they cannot come to consensus, and groups will tend to split. And so we've become very interested in trying to understand decision-making within these types of systems. Because this occurs across the spectrum, from cells to ungulates to birds and so forth. And we still have very rudimentary understanding of what's going on. It's not just cells and animals that have to come to consensus. What about humans? Consensus decision-making has emerged again and again, independently with different human societies. We are familiar with the concept of having conflicting interests. Anyone that's sat in a committee that has to come to a decision, for example, about a job applicant, is aware that the cost of not coming to consensus is greater than having this conflict among the members of the group. So failing to achieve consensus can be very costly. Individuals may be susceptible to very strongly opinionated individuals within groups, those who are very vocal about their opinions. And what about uninformed individuals? Certainly we have a feeling, and I certainly haven't found any experimental data to support this, but there's a general feeling in society that uninformed individuals are susceptible to manipulation.